In this lesson, we are going to dive deeper into the retina and focus specifically on the photoreceptors. Photoreceptors are the cells of the retina where light is transformed into neural signals to be processed by the brain. As we talked about before, there are two types of photoreceptors in the retina, rods and cones. We have many more rods than cones. There are about 120 million rods in each eye, as opposed to only about 6 million cones. Rods are also very, very sensitive to light, 500 times more sensitive than cones. Their sensitivity makes them useful for our nighttime vision. They don't need as much light as the cones do, so they are perfect for conditions in which there isn't a lot of light. They dominate our scotopic vision, which we will get into in a moment. Rods are also very good at detecting movement, but they don't process color like the cone receptors do, and they result in poor visual acuity, which is the detail in what we see. So as mentioned, we have much fewer cones than rods in each eye, only 6 million cones in each. Cones dominate our photopic vision, which is used for situations in which there is a lot of light. Cones result in high visual acuity, meaning that we are using our cones when we are seeing in detail. And cones are also responsible for our color vision. The distribution of rods and cones is not equal across the retina. The cones are primarily concentrated on an area of the retina called the fovea. What you look at and focus your eyes on falls directly onto the fovea. And this is good because this is where our cones are concentrated, and they help process detail and color, which we need to have when processing something we are focusing our eyes on. The portion of the visual field that falls on the fovea is about the size of a thumbnail at arm's length. So if you hold your arm out straight in front of you and look directly at your thumbnail, the image of your thumbnail will just about cover the fovea in each of your eyes. That is the size of the visual field that falls on the fovea. The actual size of the fovea in each eye is about the width of the lead in a pencil. And you can test out the fact that your detailed vision is focused in this tiny portion of the retina by trying something such as reading out of the corner of your eye. Without directly looking at the text you're trying to read, it is pretty much impossible. The text is still falling on the retina, it's just that the portion of the retina receiving the text doesn't have vision with high enough acuity to be able to read. Interestingly, some animals have two foveae, such as some birds of prey. This could be helpful for being able to see in detail straight ahead and down below simultaneously. In this figure, we can see the distribution of photoreceptors across the retina. Along the x-axis along the bottom, we have the location of the retina, and along the y-axis along the left-hand side, we have the numbers of receptors per square millimeter. Looking at the x-axis, 0 degrees is where we would find the fovea, and to the left and right of that would be to the left and right of the fovea in the retina. Shaded in gray, we have the cones, and you can see overall that there are many more rods than cones. However, right at the fovea, we have all cones and no rods. Then, as we move away from the fovea into the periphery of the retina, the proportion of cones to rods quickly drops, so that we have many more rods than cones in the periphery. As you might imagine, rod receptors are much better suited for viewing stars at night than are cones, since rods are more sensitive to light. However, since our foveae are comprised of all cones and no rods, it can actually be more helpful to view stars from our peripheral vision than by looking directly at them. By looking slightly to the left or right of a star, the light of the star is more likely to be picked up by our rods and will therefore be brighter. You may have noticed in this figure here that at almost 20 degrees to the right of the fovea, there is a section of the retina with no receptors at all. This is the blind spot. the location of the optic disc, where the fibers of the cells of the retina exit the eye, as we discussed in a previous lesson. Duplicity theory is the idea that humans have two main types of vision, which can basically be described as a vision for daytime and one for nighttime. Photopic vision describes our daytime vision, or for well-lit conditions, when our cone receptors dominate. With this type of vision, we can see in detail and in color. Scotopic vision describes our nighttime vision, or for dimly lit conditions, when our rod receptors dominate. With scotopic vision, we only see in black and white, and our acuity is low. However, this is when our vision is most sensitive to light. There is also a middle ground between the two, referred to as mesopic vision. This vision occurs when we are transitioning between photopic and scotopic vision, when both cones and rods are active. We use this type of vision during dawn or dusk, as one set of photoreceptors is slowly taking over for the other. In this figure here, we can see which wavelengths of light we are most sensitive to at each type of vision. In other words, it shows which colors appear the most bright or intense at each type of vision. During photopic or daytime vision, blues and violets are darkest, and yellows and greens are brightest. During scotopic or nighttime vision, reds, oranges, and yellows appear darkest, and greens and blues appear brightest. The rods, active during scotopic vision, don't actually detect the hue or color, but those wavelengths will just appear to be of a brighter light. So as vision switches from cones to rods, or from photopic to scotopic vision, we become more sensitive to the shorter wavelengths of the spectrum. This shift is referred to as the Purkinje shift. You can observe this if you pay attention, for example, to flowers in a garden. During the day, yellow flowers will appear lighter and brighter, and as night falls, the greens of the foliage and blue flowers will appear lighter and brighter. Dark adaptation is a process of our eyes becoming increasingly sensitive to light as we are in a dark environment. As we have talked about, one way in which our eyes adjust to dimly lit conditions is by pupil dilation, which will let in more light into the eye. But dark adaptation is different. Dark adaptation goes further and involves a process in which the photoreceptors of the retina, the rods and cones, become more and more sensitive. Through this process, our eyes reach their maximal ability to detect light. Dark adaptation involves a two-stage process. Immediately after we enter a dark environment, both the rods and the cones begin to enhance their sensitivity to light. The cones achieve this a little more rapidly, and within about 5 minutes, they have maxed out their sensitivity. Up until this point, the cones are more sensitive than the rods, and they are therefore more in control of our vision than our rods are. However, once the rod's sensitivity begins to surpass that of the cones, referred to as the rod-cone break, the rods take over our vision. By about 25 minutes or so, our rods reach their maximal sensitivity. Unfortunately, as soon as your dark adapted eyes are exposed to bright light again, you lose this enhanced sensitivity to light and need to start all over again to regain it. There's an interesting theory of how pirates and other sailors may have been more likely to wear eye patches to preserve dark adaptation in at least one eye. The theory goes that since they would need to go from the brightly lit ship decks down into the belly of the ship below the deck, and waiting about 20 to 30 minutes for their eyes to adjust to the darkness wasn't really an option, that they would wear a patch over one eye to maintain dark adaptation in that eye. Then they could quickly go below deck, uncover the patched eye, and tend to whatever they needed to quickly. Visual transduction is the process in which light becomes a neural impulse for the brain to interpret. Light changes the properties of the photoreceptors and causes them to generate electrical signals. This is basically what is happening whenever our eyes are exposed to light, and helps explain why we lose our dark adaptation once our eyes are exposed to bright light. Photoreceptors contain photopigments embedded within their membranes, and when these photopigments are exposed to light, they change their shape, and this change in shape initiates visual transduction. These photopigments consist of long strands of protein called opsin with a single lipid molecule called retinal attached to it. Each visual pigment molecule only has one retinal molecule attached to it, and it is this retinal molecule that is sensitive to light. When this retinal is exposed to light, it breaks off the opsin and causes a change in the activity of the photoreceptor, which results in a neural impulse. This process of retinal being activated by light and separating from opsin results in what is referred to as visual pigment bleaching. What we can see here is a schematic of a frog retina as the photopigments it contains become bleached. The figure on the left is what it would look like moments after the retina was exposed to the light. It is still deep red due to the high concentration of visual pigment that is still unbleached by light. This unbleached visual pigment still has its retinol attached to its opsin. As the retina continues to be exposed to the light, the pigment becomes more and more bleached due to the retinol detaching from the opsin. Fortunately, once a retinol molecule splits from an opsin molecule, it immediately begins to reattach to opsin in a process called visual pigment regeneration. Otherwise, once all of our retinol separated from the opsin, we would be blind. This actually can happen. Individuals who have suffered a trauma to the eye, resulting in a condition called detached retina, have the problem that their retinol is not able to rejoin with the opsin, and they become blind in those parts of the retina. Under most light levels, we always have some photopigments undergoing bleaching and some that are regenerating, so we never have to worry about being temporarily blind. When we enter a dark environment, the proportion of photopigments undergoing regeneration versus bleaching increases so that eventually most of our photopigments are in a non-bleached state. This is why our eyes become more sensitive to light during dark adaptation. The reason it takes rods longer than cones to reach maximal sensitivity is that it takes rods longer to complete the process of regeneration. Interestingly, retinol is synthesized from vitamin A, or beta-carotene, which is why you always hear that eating carrots will improve your vision.